Okay, good afternoon, everyone. So I'm Peter Hoed, PI from the European project EXIMIUS, and I'm happy to have so many people joining in for our symposium, which is on emerging contaminants, health risk associated with microplastics and indoor air quality. So first, some small issues concerning the practice of this Zoom session. So for your notice, this event is being recorded, and we will share after a week or two this on our EXIMIUS website and on social media challenge. So if you do not want interventions, if any, to be shared, please let us know on our info email address info@eximius.eu. So other practicalities, you can see at the bottom of your Zoom screen a notice for the chat at one hand, more on the left normally, and a notice for Q&A. So these are two possible ways to communicate because the voice is not open for the people listening in, only for the presenters. But these are two ways to communicate. We would like to use as much as possible the chat box only for technical issues. So if something happens that the slides are not visible, you cannot hear us, please let us know about the chat so that we can work on it as efficient as possible. Then the other box, the Q&A box, a bit more on the right, is meant to be used for scientific questions concerning the talks you hear, concerning the issue, microplastics and indoor air. And we try in the Q&A session after each talk to bring these questions to the speakers. But if there are many questions, we can also try and answer these questions via this chat box. So keep your eye on that. If some questions could not be fully answered orally. And then, Joe, just a quick view on what is our European project EXIMIUS. So it's a cohort study, or different cohorts that we study, and we try to look in the human exposome with a focus on the association of the human exposome and immune-mediated diseases. So here you find a cartoon that you also find on our website. So it is a program funded by Horizon 2020. It will run until the end of June 25. So originally the end was foreseen by the end of the year, but due to, as many projects have had difficulties in the COVID period to sample, and that's very important in epidemiological studies, to sample in the field. We did ask and did get an extension of 6 months. Our total budget is 10.8 million euros. We have 15 partners in 7 different countries. And our project is also integrated in the human exposome network. So here you see our 15 partners, so spread over different locations in Europe, as you can see, with universities and 2 small SMEs or non-profit organizations. And then this is an overview of the different exposome projects being embedded in the cluster of the European Human Exposome Network. So it is a different exposome projects of which us is one of them. Of our project is one of them. So these projects, they have all a different focus point, and all together we collect quite some data on the human exposome, internal and external exposome, and human health. And you can find more information on this at www.humanexposome.eu. And so, coming back to symposium, so this is the 7th symposium we organize within the framework of our project. I will not read the titles, but we had 6 earlier ones and you can find them more or less in full on our website. So if you want to see lectures that have been, you can still have a view on that. And so you can find that on our website, EXIMIOUS-H2020.eu. And so coming back to today, so the emerging contaminants, microplastics, and indoor air quality and health risks. So here we are lucky that we have 3 speakers in the field. So Dr. Raymond Pieters from the Netherlands, Xavier Basagaña from Barcelona, and Hubert Dirven from Norway. And so I will then introduce our first speaker. So that's Professor Raymond Pieters. He will talk about the human risk assessment of microplastics. He is an associate professor in immunotoxicology, working in innovative testing systems and involved in different projects. And yeah, his domain of interest, quite logic, I think, seeing what he is doing, is the effect of different substances, drugs, pollutants, microplastics on inflammatory processes and immune responses. So I will stop sharing my slides and I give the floor to Dr. Raymond Pieters. And I will turn off my video, so then Dr. Raymond Pieters can, or you can focus on his talk.
Yeah, okay. Can you see my slides and hear my voice?
Yes, very clear.
Okay, great. So first of all, I'd like to thank the organizers to invite me to present polyrisk, in particular, the project I'm leading on human risk assessment of microplastics. You see all the partners involved, among those is Hubert, so NIPH, Norway. So we work together closely on this project with all the other partners. And I'm also linked to the Momentum project, which is a Dutch project on microplastics. And what I would like today is to show you where we are now, what we have done in the last 3.5 years. So the project is lasting another half year, and then we should at least have some answers to the questions. And the questions, of course, in this is what is the risk of exposure to microplastics for human? And of course, you know, this one is a very familiar formula, I would say. So the risk we are interested in is a combination of hazard and exposure. And in particular in polyrisk, we have a lot of attention to exposure, which I will show you later. So we look in particular at effects on the immune system, because that is something we can relatively easy measure in humans. And we also have a lot of assays that allow us to check the effects of particles, in particular these particles on immune cells. So I would like to start off with what we have actually as one of our results of polyrisk is to design a risk assessment framework. And I'm showing this because we have a sort of decision, well, we came up with a sort of framework that gives us some decision notes to assess the risk of these particles. And this is just one example of a risk framework, but in particular for human exposure via the lungs. So what we start off with is that we look whether microplastics are really inhaled. And if they're inhaled, we know, or we can at least estimate where the particles end up in the lung. And if they are really small, they end up really deep in the lung. And then we actually know what we should measure, because then the particles are in the alveoli, where they encounter macrophages and epithelial cells. So that is a sort of framework we use. In the framework, you also see that there's a distinction made between, for instance, fibers and polymers of low concern. So the fiber, we know actually what to do. There's a regulations for fibers, so we can group these fibers. Okay, we can at least sort of estimate the risk of these fibers. More complex is the MNPs we are interested in. These are MNPs, micro nanoplastics of very small sizes, usually. They may have all kinds of substances on their surface, etc. So then much more should be measured, and we don't have the tools yet available. So the challenges are really in testing these polymer-derived particles. And this slide actually shows you the complexity of the subject. And actually, I think it's a nice picture where everything comes together. So what you see here is an individual that can inhale or ingest particles. Maybe there's also some dermal exposure, but actually we don't have much proof for that. Most of his particles are probably inhaled. And as you can imagine, these are particles. So we know something about particle toxicity, and we know that these may induce oxidative stress, induce inflammation, even translocate, etc., may in some cases induce cancer. So we have knowledge about particle toxicity, which may also apply to micro nanoplastics. But the additional issue with micro nanoplastics is that it's not only polymers, but these microplastics include many, many additives. And I think Hubert later on will talk more about this, but these additives may also have certain effects, ranging from liver toxicity to endocrine disruption. So that makes microplastics more complex than, for instance, nanoparticles that are designed to be nanoparticles. So this is something, the additives, but on top of that, these particles, as also airborne particles that we already know of, like diesel exhaust particles, carbon black, etc., they may also absorb contaminants from outside, so polyaromatic hydrocarbons, for instance, but also pathogens or parts of pathogens, microbial components. So, and all this makes it really an exposome-like topic from the start. We don't only have to deal with the polymers per se, but with many more chemicals that are in the plastics or adhere to the plastics. So this is actually the complexity. We have the particle toxicity, we have chemical effects of all kinds of chemicals within the microplastics, and on top of that, we also have to deal with microbial effects. So I would like to show you some earlier studies we and others have done, but I will focus on the studies I have done in my lab. So we did do some research on mice and humans to check for the hazards and risk of airborne and nano-sized particles, in particular polystyrene particles. So this is a study that we did a while ago. It's almost 20 years ago, but it's still valid, I think. What we did, we exposed mice to carbon black. And that's what you see here in the lungs. If you give intranasal exposure, you see these particles end up in the lungs. And if you look at these macrophages and neutrophils, you also see that they contain these little black particles. This is carbon black. And what you can see immediately after exposure is that there's an increase of all kinds of leukocytes in the bulb fluid, so in the bronchoalveolar lavage fluid. and these contain neutrophils, eosinophils, etc. On top of that, we have a protein release in the bowel. We have TNF expression, and we have a sort of angle, side scatter analysis it is. So you have a kind of inflammation very shortly after exposure. Interestingly, or importantly, I would say this is very high dose. It's 200 microgram per mouse, which is enormous if you compare it to human. And if you go 10 times lower, we don't see anything anymore in these mice. So it shows you actually that this is actually a reflection of what we call the overload hypothesis. So, and that's a hypothesis raised by Günther Oberdorster a long time ago, and he said, well, only in overload conditions you will have these immune effects. I will get back to that later. We also did polystyrene in these days, and this is polystyrene with different sizes, also with a high dose. So you have the 50 nanometer up to 1 micrometers. And here's the 50 nanometers, and you see that these polymers in the lungs really cluck together. And the smaller the sizes, the more particles you, of course, give to the mice, and then you see these enormous effects. And again, we need a high dose to get these effects. Again, this is to illustrate the high dose that we need. And on top of that, in both cases, so in case of polystyrene and in case of carbon black, we also find in these mice that there's an increase of an immune response later on. We find an IgE response against a co-administered protein. So these particles may elicit or induce allergic responses. And that was in mice, not in man. We also did human studies in the past and focused on air pollution exposure. And what we did is a study that we also did in polyrisk, but this was not before the MNP hype, I would say. And this is done just to check airborne pollutant effects on immune system of these volunteers. And we did several locations. Here you see them in the Netherlands. And we did, for instance, a stop-and-go and a traffic situation. And this, in fact, is in the Schiphol underground. And what we found is that 2 hours after exposure, so what the people were exposed to is just air, and they cycled on the spot. And after 2 hours, we found an increase. If we looked at all sites, we found an increase of neutrophil in the blood. And 18 hours later, that was normal again. So there was a temporary increase of white blood cells, in particular neutrophils. And that indeed showed us that exposure to particles in the air would induce an inflammatory response. We didn't check for microplastics, but they were included, I guess, because that's what we measured later, and I will show you later the recent results. But this already indicated that particles can induce an inflammatory response in mice, but also in humans, and in particular at certain concentrations. Just one step aside, this is one slide, and actually there will be one follow-up on the immune system, just to give you a flavor of what we look at in these studies. So we are interested in inflammation, and inflammation is mostly driven by the innate immunity. And the innate immune system is composed of epithelial cells, but also neutrophils, macrophages, etc. And I will focus on, in my presentations, we will focus on neutrophils and macrophages, and on top of that, on dendritic cells. And these dendritic cells, as you may know, translate innate signals to the adaptive immune system, and the adaptive immune system will produce antibodies or T cells. And long-term, for instance, allergies, of course, are not depending on the adaptive immune system. With the knowledge we now have on inflammation, we recently developed an AOP for inflammatory responses, and we identified 3 key events for inflammation, which is tissue resident cell activation, increased release of pro-inflammatory mediators, and leukocyte recruitment or activation. So these three key events we have taken along in our project. And for that matter, we look, for instance, at activation of epithelial cells by micro nanoplastics, or activation of macrophages, and release of inflammatory signals, including cytokines. So what we did actually, we tested many particles, or many for actually microplastics. We also tested titanium oxide, and as a sort of positive control for the immune system, we also we compared our data with LPS, which is a component of gram-negative bacteria. So what you can see here, if we expose mitochondria, which is a THP1 macrophage, so a cell line that can be developed into macrophages, we expose these cells to different particles, and what you can see is that it's a relatively high dose, again, 100 microgram per ml, you see a decrease of mitochondrial activity and an increase of LDH leakage, and that means that the cells are disturbed or a little bit, they feel not so happy, I would say. Again, keep in mind this is a relatively high dose, and what we didn't find and still expected was an increase of NF-kappa B. NF-kappa B is one of the transcription factors for a lot of cytokines, but apparently these particles do not stimulate this pathway and none of the dosages or concentrations. And of course, the control, LPS, is clearly capable of doing so. Also, we didn't find cytokines. So it illustrates, these are all primary particles. So from this, we assumed more or less that primary particles freshly produced are not so active on in vitro assays. Keep in mind, this is all in vitro assays. Nevertheless, these particles were taken up. We found it for all particles, but we tested for polystyrene, which you can buy fluorescently labeled, so we can identify them in microscopy or in flow cytometry. And if you look carefully, for instance, you can also focus on the graphs below here, which is a reflection of these flow cytometry data. You can clearly see that the small particles are taken up in every dose, and also very fast. Within 15 minutes, at a certain concentration, all cells were positive, and after 3 hours, it's clearly positive. The higher particles, 1 micrometer particles, are taken up also, but with a slower kinetics. The other particles that I showed you before were not fluorescently labeled, would be identified that they were taken up by looking at the lysosomes. So we saw an increase of lysosomal activity, which points to the fact that they are taken up. So nevertheless, the particles are taken up, and then nevertheless, they don't stimulate an increase of cytokines, although they cause some damage to the cells. How to translate that to in vivo is not so easy, but that is probably due to the fact that what we inhale, what we are exposed to, are not primary particles. They are weather particles or H particles. We also tested this in neutrophils, freshly isolated neutrophils. So keep in mind that the neutrophils were increased in these human exposure studies. But also here, we didn't find really much, except for if we use a very high dosage. So here you see the viability of, or actually the annexin 5 expression, which means if it's higher, then there's more dead cells or dying cells. So here you see that a high dosage of 1000 microgram per ml in vitro, you have an increase of these dying cells. Well, if you go lower, 100 microgram per ml, you don't see much. And the same is for PVC. High dose is doing something, lower doses. Except for these particles, well, we in our project we have produced particles ourselves. That's done by Corina Altmann from the BAM in Germany, and these 2 particles appear to be positive. This is polypropylene and polyethylene, and they show an increase of NF-κB activity, which can be only partly inhibited by polymyxin B, and polymyxin binds to LPS, among others. So if you see and decrease with polymyxin B in case of polypropylene here, it might be that LPS is part of the part of the cause of the increase. We'll still work on this because we're not sure how it really works, but it appears the chemistry people in our project have identified oxygen groups on the surface of the particles, and they are now try to identify which type of oxidated groups we have to take along here. But anyway, these are positive particles, and they are not, I would say, not primary, but they are secondary because they are treated in a way that they have become positive. Okay, this is the in vitro work. This is another one. This is about polystyrene particles, and this was actually, it also illustrates that in particular weathered or aged particles are more positive than the naive or primary particles. You see here that the particles are all taken up, irrespective of the size. So you see different particles here in the dendritic cells. And these dendritic cells are monocyte-derived dendritic cells, so human dendritic cells. And if we expose them to the particles, only the weathered particles, so what we actually did, we exposed polystyrene particles to water of the River Rhine for a month, and then extracted the particles again, exposed it to dendritic cells, and looked what happened to these dendritic cells. And what we see is an increase of these molecules, CD83, and these are molecules on the dendritic cells that are needed for activation of T cells. And indeed, if we combine this with T cells that are normally not activated by these dendritic cells, if we have this stimulation of these dendritic cells by the particles, then we see an increase of this T cell response. So here is the T cell response only with the weathered particles and only with the smallest one, although the intermediate size also does something. And this illustrates that something in the environment has changed the particles so they are active to the immune system. So again, primary particles seem not so active on the immune system, while secondary and weathered particles may be positive. And that also links to our real world scenarios. So in polyrisk, we have done several real world scenario studies in which humans were tested and exposure was tested. And I will only show the urban outdoor ambient exposure scenario where individuals were exposed to traffic exhaust. And the study was like this. We had young volunteers, and they were sitting on a bicycle for 4 hours. So a static bicycle, 20 minutes per hour. And they were on different locations, one stop and go, one along the highway, and one in an urban park. So this is the setup. We took blood at 6 hours, at 17 hours, and analyzed the exposure. So we collected airborne particles, and we also collected blood and saliva, etc. So with that in mind, I'd like to sum up. We really have, with micro nanoplastics, we are dealing with a very complex challenge. It's not a simple chemical, as you know. Particularly for hazard assessment in vitro, it's very complex. If you don't, we really have to also consider the chemistry to really understand what's going on. Exposure assessment is really difficult. We don't have methods available yet to quantify the number of particles. We can, what I showed you earlier, we can do blood analysis and also analysis in airborne particulates, but that's all with pyrolysis GCMS. Counting the particles is still very difficult. So that also hampers proper risk assessment, although we do an effort, as I showed you. But it's a challenge. And as said, primary microplastics do not have a very strong effect on the immune system, at least not pro-inflammatory. It may, at high dosages, decrease the cell viability. But that's something that's not so strange. I mean, you overload the cells. And we think that weathering So particularly leading to altered surface chemistry, maybe a release of adhered chemicals or leachates. They appear to render some weathering is needed to make MLP more effective. But nevertheless, human data indicates that exposure to tire wear particles, and these are not primary microplastics, but secondary, may have an increased effect on the amounts of blood cells. So here's the team I like to acknowledge. You see me here, but also Hubert, who will take over in next presentation. This was 2 weeks ago, I think. Yeah, no, last week. Yeah, in Bucharest. And I also like to acknowledge CUSP. So we are not on our own polyrisk. We have a good collaboration with other CUSP projects to make this a success. And hopefully you like my presentation, and I'd like to thank you for your attention. And I stop here.
Thank you, Raymond. I have a look to the Q&A. I don't see a question popping up yet, but let me maybe ask first a question. So very intriguing human data. And yes, certainly other materials are also there in the air that people inhale. But it was intriguing that the PM10 had a very low value, I did see in this graph. But do you have any idea, or is there any interaction with the rubber particles and, yeah, let for instance, irritating gases such as nitrogen dioxide or anything? Do they play together, or is that really a separate? Can you easily separate out?
No, you're right. That's also something we are now testing, of course. I mean, but what I can say at least is that the monocyte increase really depends on the tire wear particles and also on the linked chemicals, so the metals that are also in the tires. But it of course also links to what we also showed is black carbon. I mean, and that's not so strange. And I don't know whether what we measure is not the carbon, but the blackness of the filters. And of course, rubber tire wear particles are also black. So it mixes up a little bit. For the PM10, I'm not sure. There's something that I cannot explain, but the link with black carbon is something we expected, although we don't see much of an effect there actually.
Yeah, but if I
can show me.
I don't have to make a private discussion, but I think, and good that you bring it up, the black carbon, I didn't think about it, but in the rubber, they add a lot of carbon black or black carbon. Huh?
Yeah, so we don't know exactly. So here's the data again, you see it, right? So the black carbon, in some case, it's a little bit increase, it's not significant, but you see some increase. We're still working on it. So, but we don't see anything on this side. Maybe some nitrogen, but ozone is not. And we also have a whole range of chemicals, which I cannot show you now. It's pHs, etc. They're also measured. But we don't see much link or correlation with exposure.
Okay, thank you. I see here a question from Ricardo.
Yeah, that's a very good one. Yeah, you're right, Ricardo. That's something we realize now that we should do. But as you know, the CUSP projects were sort of first exercise to look at microplastics, and we at that time we thought we can do a short-term study, but to look at what the effect would be on something like allergy or an inflammatory response, we need to have a much longer exposure regimen. That's absolutely true. That doesn't make it more easy. But even more complex, actually.
Yeah.
In Momentum project, other people work on the link between existing human data from a cohort study that we have done and that has been done in the Netherlands, PYAMA study. And there they also see a link between, and it's still work in progress, so I need to be careful again, but I see a link between polymer in the blood and cell effect on the immune system. But that's very, very preliminary.
Okay.
Yeah, Yuli, very good. We prepared it in a way that we took polystyrene. We bought polystyrene from a company and added with a river Rhine water for a month in shaking conditions with UV light, etc. So it was really as much as possible environmentally relevant. Then extracted the particles and used them for our studies. What we do know is that LPS is here also involved. So what we try to do is to block the response with polymyxin B, and with polymyxin B we could completely reduce the effect of the weather particles. So in this case, it was really microbial contamination.
Okay, yeah. Thank you. I had a question that links into Ricardo's question also, because you showed there in the beginning this framework for microplastics risks and the microplastic fibers as they are. Yeah, there is a fiber legislation. But do you have any clue for different plastics? What is the half-life time in the lung of fibers? Is that anywhere known?
No, yeah, not for me. I mean, all the kinetic studies are also studied nowadays, but there's no data yet. But I know people are working on it, try to get some kinetic data. I can only tell you from the mouse studies. So we did carbon black, titanium oxide, polystyrene, and different sizes of particles. And after, well, we could only follow mice for 28 days, and those particles were still in. So they couldn't get out, although in these mice, we also saw that the particles translocated to the lymph nodes of the lung. And that is something you particularly can see with carbon black. You see these lymph nodes turn out a little bit grayer. So we know that translocation will occur in that direction. And maybe if, I don't know, it's all guessing. I don't know.
Yeah. No, but yeah, having any knowledge on the half-life time in the alveoli.
Yeah, and that's very important. And also what we learn now, if you look carefully in the literature, you see that some polymers can be broken down by enzymes, and others not. Mm-hmm. So it's a wide field with a lot of knowledge that is evolving at the moment.
Very
we also saw you can find the literature if you ingest particles, they may be already oxidized. So there is something going on even in your gut. The lungs is of course different, but I would expect that there's some change in microplastics in these environments, but we don't know yet.
Mm-hmm. Okay. I see maybe a final question here in the Q&A. So from Tina.
Okay, yeah. What we see now, until now, and that's just still the stats that are being done, the statistics, it appears that it's all linked to the effects we see on monocytes and also neutrophils. It's linked to the polymers. And some link with these metals, as I showed you. But we didn't find a link with LPS. Good question. I think we didn't even check LPS contamination. So that's a good question. But
Yeah.
allergen binding, I don't know. I mean, it's also very direct. So the people on the stop and go were immediately exposed to the particles. So the particles break and start again, and that was the location. So I think there's not yet any binding of allergens, although of course I'm not sure.
Yeah.
And for the park, I would say, well, there's hardly any effect in the park, although there's also that this is a urban park, so there's still cars circling around the park. So it's not that it's non-exposed; it's low exposed.
Okay.
But I don't know. Sorry, I don't know.
Good. Just to wrap up, maybe I just think about it. If it's cars and rubber breakdown. So electric cars, they do not have exhaust, but they seem to have a larger offset of rubber because of acceleration and so on. So do we need to be worried?
Yeah, to some extent, but we will not have flying cars soon, I guess. But on the other hand, I know that the industry is working on better rubbers. So I think the development should be there that there's less tire wear, I would say. There's also something like brake wear, but they already made devices to collect the brake of the wear of brakes. So I don't know. I mean, of course, we cannot drive without tires.
Yeah.
But on top of that, it's good to realize, and that's something we also have in the institute and work in EOS, we discuss this often, is the microplastic particles issue. At the moment, it's only, let's say, 1 to 2% of all the particles in the air. So it's really, really low at the moment.
Yeah.
So at the moment, I would not worry too much.
Okay.
And it's a matter of risk benefit, I would say.
Mm-hmm. Okay.
So there's a risk if you are next to a stop and go, and maybe if you pass the stop and go every day, then you will have more microplastics from tire wear. But in comparison to airborne pollutants from car exhaust, I'm not sure.
Yeah. Okay. Thank you. But I just read in the chat that people cannot read the questions. I did not realize that too well. So the last question was clearly about the rubbers and so on. There was an earlier question on what was the water about, and that was also clearly answered, I think. And now the questions are removing, so I cannot read them anymore.
Apologies.
Okay.
The questions are first shown to the panelists, and once they are answered, we have to actively click on them. And now I'm making them available so that everyone sees them in the Q&A.
Okay, very well. So I will share my screen for a second because I have to thank again Dr. Raymond Pieters, a very interesting talk, very intriguing data. So thank you very much. And so then we come to our next speaker, and that is Xavier Basagaña, who will talk about air quality and children's cognition with a focus on school air quality. So he is a biostatistician and working at IS Global as a professor and coordinating different projects there, H2020 projects. So his field is mainly statistical methods and then linking that or verifying that with the observation in studies. So, and mainly air quality and health. So I will stop sharing and so then Professor Xavier Basagaña, will take over. So please, I can see your slides. That seems to work. I do not hear you. Yeah, now I hear you.
Okay. Thank you. Yes, my talk is a bit on a different topic. So I'm going to focus on air quality and the relationship it has with children cognition, with a focus on school air quality. So, well, we all know that air pollution has multiple health effects, and probably the ones that are most widely known are the respiratory effects and the cardiovascular effects. But now for a long time, there has been many other studies linking air pollution with many other outcomes: diabetes, premature birth, decreased birth weight, and several are relating air pollution with the brain, and the concentrations of air pollution may influence neurological development, also mental health, and even there are some studies that are linking air pollution exposure with neurodegenerative diseases. But we knew for a long time that some chemicals are neurotoxic. So, for example, with the example of lead, we know that for a long time. And it all started with poisoning episodes after being exposed at very high doses. But then by 1959, there was already a concern about what the airborne concentrations of lead could do, and if they had health effects or not. And this led to basically 30 years of research until lead in gasoline was banned in the US. And all this research, well, one of the things that was reported in this research is that the children's brains are the more sensitive target. So more recently, there has been a lot of other epidemiological and toxicological research that have been investigating the link between ambient air pollution and outcomes on the central nervous system or on the brain. And for example, this in this paper in 2012, that this is a result of a workshop on air pollution and brain health. This paper was already discussing much of the evidence that existed at the time, linking air quality or air pollution with chronic brain inflammation, microbe activation, lower IQ, neurodegenerative diseases, autism spectrum disorders, and other outcomes. And then in 2016, there was this other study that I think that also made a big step forward in the evidence that actually air pollution can damage the brain, offering new insights. So they were able to detect particles from combustion. These nanoparticles, they were able to locate them in the human brain. And these particles can enter directly through the olfactory bulb. And once they are there, we know that they are toxic, and because they can produce damaging reactive oxygen species. So this adds to all the evidence that we had before. So what I will try to do in this talk is I'm going to describe some studies that have investigated the link between ambient air pollution and cognition in children. I will mostly talk about studies that we have conducted in IS Global, not because I think they are the best studies, but because they are the ones that I know better. And I'll try to give a focus on school air and indoor air, some results related to that, and we'll briefly go through some of the challenges that we have with indoor air. So I'll start with some results of the BREATHE project. This was an ERC project by Professor Jordi Sunyer, who has been a pioneer in this field. he and his team conducted this study that involved almost 3,000 children aged 7 to 10 from 39 schools in Barcelona. And what they did was to measure the air quality in the schools, both in the classrooms and in the playground. And then in parallel, they also evaluated the memory and attention of the kids. They did that during 4 times, so every 2 months during an academic year. So they ended up having a lot of data, more than 10,000 tests. Just to give a brief description on the air quality of these schools, here you have different pollutants, elemental carbon, NO2, and ultrafine particles, indoor and outdoor concentrations. And if we look at the median, and maybe in at NO2, which we may be more familiar with the levels, the outdoor concentrations were 48. And just for reference, in the background stations of Barcelona, the average was 41. Actually, what happens in the city of Barcelona is that schools are very close to traffic, and that's why they have high concentrations of pollution. This is outdoors, and indoors also they had around 30. In the study, we also measured PM 2.5, and we did source apportionment of this PM 2.5, both on the outdoor and on the indoor measurements. And if we start looking at the outdoor measurements, we see that what is contributing the most to PM concentrations in the schools was the mineral source, and this is mainly due to schools that had sand in the playground. The second one is already traffic, and then there are other sources. But if we look at the indoor PM, we see that the main source here is this one that includes chalk that they use for the blackboards, and then organic that is mainly from the skin of the children, and textile, which is from the clothes. So the major source of indoor PM 2.5 was this. Then it came the mineral source, also mainly driven by the schools that have sand in the playground, and then traffic. So this already indicates that using PM 2.5 may not be a very good marker if we are interested in investigating the effect of traffic related air pollution, because there are many other sources that are contributing, and some of them, they do it with a greater amount than traffic. There were also some studies looking at the infiltration of these pollutants. So you have the indoor concentrations in the y-axis and the outdoor concentrations in the x-axis. And you see that, well, there is a correlation for most of them. Well, for NO2, concentrations are higher outdoors, but there is a correlation. There is some infiltration for elemental carbon, and there is very good correlation. So it's a high infiltration from the outside in the classrooms. For ultrafine particles, well, they're clear. There are other sources that don't come from that do not come from the outside. And here, for a marker of breakware, there is also some correlation indicating infiltration from the outdoors. In the study also, we conducted a sub-study on personal exposure. So some children were wearing blood carbon monitors for 48 hours. And at the same time, they were filling a time activity diary every time that they changed the location or the activity that they were doing. So with that, we tried to calculate the personal inhaled dose using inhalation rate factors depending on the activities. This was taken from the literature. So we ended up with something like this. So for each participant, we could have the total concentration, and we can estimate what percentage they got from commuting, from being at home, or from being at the school. So these are individual subjects participating in the study. And in the bottom, we have a summary table where the first row is the percentage of time that they spend in that location. And the last one is the percentage that that location contributes to the daily dose. So there are different things that we can see here. So for example, well, most of the time that these children spend at home sleeping, so 41% of the time they are there, but this only contributes to 20% of their total exposure. While, for example, if we look at commuting, they only spend 6% of their time commuting. But this contributes to 20% of the dose, because during that time they are very close to traffic. And also it's interesting to see that they spend quite an amount of time at the school, and actually the contribution to the dose that we can allocate to the school is higher than the time that they spend there. So from this we can see that both the concentrations at the school and at the time during commuting have an important impact on the personal exposure of these children. And this is the main result of the project in terms of the effects of pollution on cognition. So you have here the results of working memory on this test that we were administering during the year. So we have the 4 visits, and you can see that the lines go up. So this is because the children are improving their working memory. This is what we expect to see, because these are capacities that they are developing at those ages. But what we saw was that if we look at the schools with low air pollution, this increase during the year was 11.5%, while in the schools with high pollution, it was only 7.4%. So they also increased, but in a slower slope. And this is after adjusting for socioeconomic variables, for exposure at home, and for several other characteristics that could influence the comparison. Also, since we had the source-specific PM2.5, we could run the analysis with the different sources. And what we saw was that only the traffic source was the one that was associated with slowing down neurodevelopment of the children, and we didn't see that for the other sources. Also, in almost 300 children, we also conducted brain magnetic resonance. And here we also saw differences when comparing the highly exposed to the low exposed children in some brain areas. And in particular, we saw effect of pollution in this brain area that actually is the same area that we see when we compare younger and older children. So, and the sign is the opposite. So it actually means that it goes with our conclusion that what pollution is doing is slowing down brain maturation. Also in this project, we investigated the short-term effects of air pollution. In this case, we were looking at attention. Attention is a capacity that changes day by day, even moment by moment, and can be influenced by the environment. So we were checking if the air pollution concentrations of the day were influencing the results on the attention scores. And we saw that there was an association. We saw that when we were using the previous day air pollution concentrations, there was a decrease in attention, the higher the exposure. And this is for short-term effects when controlling for the long-term effect, which is much stronger. So you see that air pollution can have an effect of being exposed throughout the year to high concentrations, but also the day-to-day variations can have an influence as well on attention, although with a smaller magnitude. So some conclusions from this brief study. One is that the indoor particulate matter concentrations in the classrooms are affected by many sources, that there is a high infiltration of traffic-related pollution in the classrooms. Also that the time spent, the time at school and the time during commuting are an important part of your personal exposure to traffic air pollution. That exposure to traffic-related air pollution can delay cognitive development, even though in this study we could not evaluate if this delay that we saw is, let's say, momentarily, but it gets compensated afterwards, and all the children end up having the same working memory. And we saw that also that short-term variations in pollution can affect attention. So now I'm moving to another study that we did afterwards, a little bit following some of the research questions of brief. In this study, we wanted to assess again the effects of the short-term exposure to air pollution on attention, but using an experimental study. We use air purifiers to investigate this association. What we did was when we were going to school, we had a group of children and we randomly split them into two groups, and they went to two different classrooms. Well, two classrooms that were similar in all the characteristics. The two classrooms had a device there that had the capacity to filter the air. But in one of the classrooms, the device had a filter on, so it was actually filtering the air and cleaning the air. And in the other classroom, we had the device, but without the filter, so it was only recirculating the air without cleaning it. So the children didn't know in which classroom they were in, and they stayed there in the classroom for 1.5 hours, or almost 2 hours, and we asked them to do different things during that time, and one of them was these attention tests that they did on the computer. So this is the kind of test that they were doing, and we were able to do that in almost 2,000 students. So we also had a lot of data. Just looking at so here I'm showing the concentrations of PM2.5 and black carbon in the two classrooms. So the red line are the normal concentrations, the ones that we saw in the classrooms that didn't have the filter on. And the green line is the concentrations in the other classrooms that did have the filter. So we had the air was cleaned. And we saw that both for PM2.5 and black carbon, the concentrations were reduced by 90%. So we had a very good contrast between the two experimental settings. And if we look at CO2 concentrations, which we know that they can also influence attention, they were similar in the two classrooms because the filter was not doing anything to CO2, which mainly the CO2 concentrations depend on the number of children in the classroom, and this was similar in the two settings. So you can see that the concentrations are very similar, and they are going up with time, as we expect, because they are in a closed classroom, and actually they can go up to very high values. But they were similar in the two types of classrooms. So these are the results of the study. This is the attention test, and you can see that the scores of the two groups were almost the same. So we didn't find any difference in attention in this study. And our conclusion is that we wrote it this way: that cleaning the air for 1.5 hours does not have an effect on attention. This doesn't mean that air pollution does not have an effect on attention, because there may be an issue here on the timing of exposure and the effect. So if we think that pollution affects the brain through inflammation, when the children came in to the study, they had already been exposed to the air pollution of the city, and they may already had the effects of air pollution. And it may be that having them in a classroom with clean air for 1.5 hours is not enough to reduce this inflammation problem. And this is something that they found in another study. This was randomized study in a chamber only with 10 subjects, but they were exposed to very high dose of diesel exhaust and then to filter air. And they saw, so when they start, they have they did this electroencephalography. And you can see that at the beginning, the two groups start very similar, but soon they start seeing this effect on the electroencephalography. And when the exposure ended here, and they were followed for 1 extra hour, and you can see that even after the exposure has finished, the effect on the brain still remains. So this could be an explanation on why we didn't find an association in our experiment. But when we think about indoor environment in the schools and how it can affect cognitive performance, there are also other factors. It's not only air pollution, it's lighting environment, and you can see that a lot of different parameters can be measured. It's indoor air quality, of course, but within indoor air quality, there are also a lot of other things that we can measure and that could have an effect on cognition. There is also, it's important, the thermal environment and the acoustic environment. And for those also, you can measure lots of measures. So it's actually quite challenging to investigate the individual effect of particular aspects of the indoor environment in schools. If we just focus on indoor environment or indoor air quality and cognition, there are many studies, and I just listed some of them here, but there are many more that have suggested associations of different pollutants with cognition or neurodevelopment. There are papers for CO2, there are papers for PM, for black carbon, for BOX, for PAHs, for NO2, for dust microbiota, for flame retardants, and we could go on with the list. And it's actually quite challenging to investigate which particular pollutants are the ones causing the effects or how they act in combination. So we need exposome-like studies to address this question. Here I'm listing some more recommendations that have been done for indoor air with a special focus on indoor air quality. This table summarizes some of the main ones that were listed in this paper, but the paper includes others. One is raising awareness about the impacts of air quality, trying to reduce the use of private cars, and promoting the active travel to the schools or public transport. Also, there were some recommendations related to cleaning. That the cleaning of the classrooms should be done ideally after school hours and opening the windows, and if possible with green cleaning products. Also important to reduce traffic in the school surroundings, and this is something that could be implemented by the city managers. Another recommendation was to clean and replace the sand from the playgrounds periodically, because some of the other particles also get deposited in the sand, and this is then resuspended. And if possible, to try to move the schools or the classrooms away from traffic. So just to sum up, the main points that I wanted to come through in this presentation is that traffic-related pollution is probably associated with cognition, that school and commuting time contribute importantly to personal exposure in children, and this is something that should be taken into account in when studying the relationship between air quality and outcomes in children. But more research is needed, especially on the interplay of different indoor air quality parameters, specifically when we're thinking about cognition, but possibly also with other health outcomes. And also there is a need on knowing more about effective interventions on indoor air quality with the final aim to protect health. And I'll finalize just mentioning that there is a cluster of Horizon projects called the Ideal Cluster, so that there are many scientific projects that are within this cluster, and precisely they are working on the topic of indoor air quality and health. And hopefully in the next few years, these projects will be able to provide new insights on indoor air quality and that's all I wanted to explain you today. And if you have questions, I'll be happy to answer. Thank you.
Okay, thank you very much. Very nice overview of two larger projects, and that really shows how important it is to look to different parameters, and that it's not always simple to really find the hazard situation or exposure. One question I had on this, the second study, I think Attention or something it was called, is then these filters that are placed, were there also non-filters placed, but still making the noise in the classrooms in the control classes? Because I could think of that noise is maybe a very important one here.
Yeah. No, they were the levels of noise were similar in the two.
Yeah.
Well, we actually measured also noise, so we were able to control for that as well.
Okay, thank you. I do not see any questions in the Q&A, and I don't know if there's anything in the chat. No. So that gives me then the opportunity to ask a second question, if I may. So yeah, the filters, they take out the particles, but as you showed, not for the carbon dioxide, but for other gases, for instance, nitrogen dioxide or nitrogen oxides. Were they reduced due to filtering? Because that they could also play quite a role, I think.
No, I think they're not. There are some purifiers that actually offer the capacity to reduce also the concentrations of some gases, but they have some chemical process. This was just a physical filter, and in principle it did not affect the concentrations of the other gases. There are some other purifiers that do that, but also I think well, they are a bit also controversial, because then they can also create some other compounds. So one needs to be careful with the type of purifier that's using.
Yeah. Okay. And I got a final question. Does your project look into different types of schools? Because you said 2,000 students. I mean, different types of schools, very modern schools. For instance, I live in Belgium, and we have more modern buildings where there is ventilation systems, and that they can have a filter system, while older buildings do not have that. And so there you could have a continuously filtering of the air, in fact, and were schools like that include?
So in the first study, we had different kinds of schools, but they were paired by having similar characteristics. We also did analysis on well, we collected a lot of data on the schools, on the type of windows, on the type the type of school, and we're able to study some differences also in terms of infiltration. But in the second study, the randomized one, basically this was very well controlled, because we had two groups that were from the same school, and we just asked them to go to different classrooms within the same school, and we did the comparison using the other as a control. So all the characteristics of the building were the same in the two groups. There was a perfect match in this case.
Okay, thank you. Yeah, very interesting. I do not see any hands raised or questions, so thank you again, Xavier. And then I will share my screen for a second. Again, so I can also announce that our third speaker. So yes, so our third speaker will talk about living in the plastic age, chemicals leaching from plastic, and possible health effects. So this is a talk by Hubert Dirven, who is a toxicologist at the Norwegian Institute of Public Health, and he is also an ERT, so registered toxicologist, and a member of the WHO Joint Meeting on Pesticide Residues. And his interest is really in experimental work, data to investigate the relationship between exposures and health effects, but also focus more on alternative methods and exposure assessment. So I will stop sharing and I will give the floor to Hubert, and you can now put up your slides and present your talk.
Very good. Thank you very much, Peter, also for the invitation to present this topic. I hope you can see the slides.
Yes,
Yeah,
we can
okay,
see and
very
I
good.
can hear you clearly.
So we are living in the plastic age. That is definitely the case, and we are using a lot of plastics. This is a figure from created by the group in Norway, the Grid group in Arendal, and they state that each person on the planet uses 45 kilograms plastic per year. And if we don't do anything, then the amount of plastic that will be produced will be doubled in 2050. Unfortunately, a lot of plastics is still not recycled, and it's mainly because of the cost of new plastic is lower than recycled plastic. And that means that a lot of the plastics is still in use or has been discarded, meaning disposed in landfills or incinerated. And plastics are really nice materials. It's not only a problem; they have big benefits as well. You know, you find plastics in medical devices, in all kinds of in cars, in packaging, especially in food contact materials. Food are much better preserved if they are packed in plastic and they are not contaminated by bacteria and viruses. So plastic have a really great benefit for society. But there are some problems with plastics. One of the things is that a recent report from a group in Norway, from the group led by Martin Wagner from the Technical University in Trondheim, they state that they did a literature search and they found more than 16,000 chemicals that are potentially can be present in plastic or are associated with plastics. And a lot of these chemicals are not regulated internationally. Only 6% have a regulatory status. A lot of these chemicals, almost 25%, are either persistent bioaccumulation or toxic. And that means that, especially the REACH system, the system that we use in Europe to regulate chemicals, that we have a closer look at these chemicals because these are characteristics that we don't want to have for chemicals. We lack hazard data for a lot of these chemicals, and of course, mixture effects is quite important because humans are not exposed to one chemical, but to a mixture of chemicals. And this report also gives some priority groups of concern: aromatic amines, for example, the bisphenols, the phthalates, the parabens, the chlorinated paraffins, and of course the PFASs, because they are also present in plastics. It's for me, it's a little bit interesting to see that D-ethyl-hexylphthalate is still in focus because in 1993, almost 30 years ago, I did my PhD on D-ethyl-hexylphthalate. At that point in time, it was because it was then recently discovered in the National Toxicology Program in the US that it caused liver cancers in rats and mice. And that was a surprise because DHP was not genotoxic. Due to a lot of mechanistic work, we can conclude that the mechanism of this genotoxicity is rat and mice specific and very likely not relevant for humans. So we are not so worried about the genotoxicity and carcinogenicity of DAP anymore, but we are worried on other effects that this compound causes. And also then, by that time, we already did some biomonitoring of DHP in plastic using factories. So that was just a side note. The chemicals in plastic was also the topic of a report from the United Nations Environmental Program, and they also say it's the amount of plastic that is produced and also the number of chemicals that are associated with plastics that we are afraid that there are effects on the human health and environment caused by these by the presence of all these chemicals in plastics. And then you can ask yourself, what are the chemicals doing in plastics? Well, a number of these chemicals are plasticizers, making sure that the plastic is flexible, for example. Flame retardants are used in order to make sure that the plastic furniture or plastic clothing is not easily set fire. Antioxidants, heat stabilizers, colors, light stabilizers. So there are a number of chemicals that with good functions in plastics added. But in addition, we have also non-initially added substances. So substances that are not on purpose put into the plastic. And these are breakdown products, and these are impurities and side products, and also contaminants from for example, the recycling processes. And yeah, these chemicals we don't know that they are there. We really have to look very carefully with non-targeted analytical methods to find out if they are present in plastics. And yeah, if we think about exposure, then these chemicals associated with plastics, well, during production, we can be exposed to these chemicals when the plastics are in use, then we are exposed to these chemicals. And also by the end of life, when we want to recycle the plastics or burn it or put it in the landfill, then we have still an opportunity that both the environment and humans are exposed to these chemicals. And these chemicals are, they are often not bound to the plastic. They can migrate and out of the plastic and lead to human exposure. And I wanted to show some data of a study. The report is in Norwegian. It's about chemicals in children from Norway. But there is a news story on our website with an explanation in English what the main findings were. This was part of the Norwegian Environmental Biobank, which is an initiative that we started in 2016. It's part of the mother child cohort that we have in Norway, a big pregnancy cohort with more than 120,000 pregnancies. From that big pregnancy cohort, we selected then 3,000 pregnant women, and we looked for chemicals in the blood of these women during the pregnancy and also in the urine. And then later on, in 2016 and 2017, we selected a number of these pregnant women, and we asked if they would donate blood and urine again. We did the same for fathers, and we did also the same for the children. And that collection was part of the HBM for Europe program. And now in this year, we have asked again to on the mothers, fathers, and children to give blood and urine to follow up and do additional analysis of chemicals. And what we see, for example, is that we measure quite a lot of chemicals, and these chemicals are often associated with plastics. For example, phthalates. There are a number of phthalates used in plastics. The most common one is diethylhexyl phthalate. Although the use of diethylhexyl phthalate in plastics is reduced, but still we can measure quite a lot of phthalate metabolites in urine in children in Norway. And we sometimes we see a difference between the girls and the boys, and sometimes there is not so much difference. Another group that we very often see in urine of children is the phenols, especially the bisphenol A has received a lot of attention in the last 5 years, and we can measure quite a lot of bisphenol A in urine. And also when we look at PFASs in the blood of children, then we measure currently up to 25 different PFASs, but the most common ones are the PFOAs and the PFOS and the PFAS6 and the PFNI. And we measure in almost all of these in the blood of the children, we measure one or more of these PFASs. That a chemical is present in blood doesn't mean that it is toxic. So we also compare the levels that we observed with the levels that are considered safe. And for example, this is the figure for the phthalates, and for example, the diethyl phthalate. We measure quite a lot of these phthalates in urine, but then if you calculate back what the exposure to diethyl phthalate is, then it's only 1% that are exceeding levels that are considered safe. But for some other chemicals, especially, for example, bisphenol A, almost everyone has more bisphenol A in urine than is considered safe. So yeah, it's chemical dependent if it's an hazard and possible toxic or not. And for the PFASs, just an illustration, almost 30% of the children had levels that are exceeding the levels that are considered safe. And then you can say, well, this is maybe something for Norway, but due to the HBM for Europe and also the PARC project that is now ongoing, a lot of biomonitoring data is collected in different countries. And Vito in Belgium did a really nice exercise to create a dashboard, and this human biomonitoring dashboard is really a nice website where you can click which chemicals you're interested in, which biomarkers you are interested in, the age group, the countries that you are interested in. You can compare different countries, and I would really like to promote that this dashboard is more often used in order to assess exposure to chemicals. It's a really interactive tool, and everyone can have access to it. But then, yeah, up to now we have been talking about individual chemicals, but we know that in daily life, and also the chemicals originating from plastics, that we are talking about mixtures effect. And the question is then, of course, what is the hazard of this exposure to low levels of hundreds of chemicals? And then, yeah, I always think that it's good to remember the Bradford Hill criteria to find out, is there a relationship between a cause and an effect, where you really should look at the strength of the association, the temporality, the consistency, the biological plausibility, dose-response relationship, the experimental evidence, the coherence, and the specificity. So you need to take a number of factors in account. And as toxicologist, I'm very much interested in this biological plausibility. Is there a causal relationship between an exposure and an effect? But to assess the health effects of chemical mixtures is quite complicated because you need to know what the mode of action is. And if it's a similar mode of action, then it's most often the dose addition is a very suitable default to assess the health effects. But in the dissimilar mode of action, then the components in a mixture, they have a toxic effect independently of each other. Then it's much more complicated. And in addition, there are a lot of other topics that needs to be addressed when talking about mixtures. It's really important to find out how a chemical leads to a toxic response, and its adverse outcome pathways is a very nice way to organizing mechanistic data, starting with a molecularizing event, and then a number of key events and key event relationships to adverse outcomes in an organism. And for example, this was we worked also on a project led by Jacob van Klaveren from RIVM about also Euromix. It was about risk assessment of mixtures of chemicals. And it shows that this AOP is really really of value when you look at the mechanism of action. And you see for this case, this is probably a little bit outdated figure because the AOP is even more complicated for liver steatosis, that we have quite a lot of number of molecular initiating events that are involved in the processes that automatically lead to a fatty liver in cells and liver steatosis. And that means that if you want to do an assessment of the hazard of mixtures, you really need to have a lot of data. You need to have dose response curves. You need to have relative potency factors. And then you can do an assessment, and that is noted down in the right side of the figure. If you have chemical A and B is the health effect, is that the sum of A and B, or is the health effect of the combination, is it the same as A and B, or is there a potentiation of the toxicity, which is shown also in this figure, or is the total effect can be less than the individual chemical. So you need quite a lot of data to do a proper assessment of the health effects of mixtures. But it's really interesting that this paper was published a couple of weeks ago in Science, and it's a paper from the group from Helmholtz University in Germany, and they did really a nice piece of work. They looked also in a number of pregnant women about what is the concentration of chemicals and which chemicals are present in the plasma of these pregnant women, and they found quite a lot of chemicals that are present at the same time in the same sample. And then they did all this very advanced analytical work, but then they also tested the mixtures in a biological assay to look for neurite outgrowth. And neurite outgrowth is really an important process in the development of the brain during pregnancy. And then it's very complicated, but I would recommend really to read this paper, because I really think it's a very important paper. But they conclude this something from nothing mixture effect. They show with the data from the samples from these pregnant women, when they test this in the neurite growth assay, that each chemical in itself is not causing any adverse effect in this on neurite outgrowth. But if you test the mixture, that there is indeed an addition of the effects, and in some cases really leading to potential hazard. So this mixture effect is real, and this study is a very nice example to show that indeed this mixture effect is present also in humans. So yeah, we at this moment we are very interested in the chemicals in plastic and the mixture effects. And currently there is this UN nations. They are also working on a global plastic treaty. This month, there will be hopefully the last meeting in order before the treaty can be signed. Because there is a lot of worry about this, the amount of plastic that is produced. And they want to negotiate a binding global plastic treaty. And yeah, it's hoped that it's finished by the end of this year. And the ultimate aim is to reduce the plastic pollution with 90%, and also reduce the production of new plastics with 30%. And Norway is really in the driver's seat for this plastic treaty, together with more than 60 other countries. They are really pushing forward to get this treaty in place. And Norway is also very interested in the banning hazardous chemicals in plastic and in hazardous products, and increase the circularity in the life cycle of plastics, and to stop the environmental plastic production. That are typical things that Norway like to have included in this plastic treaty. So then I would like to conclude that we have plastics, that they are an environmental hazard. They are very toxic for birds and fish, and they are also possibly a human hazard. For example, what Raymond Pieters earlier presented, there is the formation of micro and nanoplastics, and widespread exposure to these micro and nanoplastics. And we need to know more about if really these micro and nanoplastics are a human hazard. Based on the biomonitoring studies, we can conclude that there is widespread exposure of humans to chemicals leaching from plastics. And these chemicals in plastics, and especially the combination of these mixtures, are possible a human hazard. And so we need to know more on the hazard of these low dose exposure to a large number of chemicals. And we need to identify the chemicals that are really hazardous and are present in plastics. And currently there is in PARC, this Partnership for the Assessment of Risks from Chemicals. We have proposed to start a new project to look at the hazards of these chemicals that can leach from plastics. And luckily a large number of other groups involved in PARC are also interested in this topic, and are interested to work together to identify these chemicals. And of course, we all hope that chemicals in plastic should be included as a topic in the planned plastic treaty that is negotiated by the United Nations. So then I like to conclude by thanking my colleagues in my group, the chemical toxicology group, and also in the food safety group, which do all the biomonitoring work that was presented in this presentation, and also other colleagues in the department, and for example also in the Polyrisk project that are interested in studying plastics. Thank you.
Thank you very much, Hubert, for this intriguing presentation. Some different issues, but at the end, coming to the mixture of it all, a little bit of something very interesting. So I see here a question from Ricardo. Thanks for your interesting talk. The concerns you have presented are also true for bioplastics or just for oil-based plastics alone, and it disappears, this question. I don't know why, but okay. So can you give your comment on that?
I must admit, I've heard about bioplastics, but I don't know a lot about them. So I must admit, this is something. As a matter of fact, today we were discussing I'm now currently visiting in South Korea, and this question came also up in a discussion we had with a group of Korean scientists. So I really have to look into what these bioplastics really are. I think they are the plastics that are easily degradable. But I have no idea what they consist of, what kind of polymers they consist of, and are they really biodegradable? That's also the question. Or are they also still a source of micro and nanoplastics? And I have no idea what kind of chemicals are used as additives in these kind of plastics. So this is a question at this moment I cannot answer, Peter.
Yeah. No, I see your point. It's a difficult one. So something new on the block. So maybe good reasons to at least have some research on that. May I ask you something about the brown paper you showed from Science? So that really looked into very little doses of different compounds, in itself not very toxic. But in mixtures and complex mixtures, then probably yes. So I missed somewhere, is that all in vitro studies they did, or did they also link that to human exposures, or did they both, or did I get it wrong?
They did both, to be honest, but they started with plasma samples from 700 pregnant women. They took 250 microliter of this plasma, and they did this non-targeted analysis to find out which chemicals are present. And they have quite a lot of database about what chemicals they observed and what the source of these chemicals are. Are they industrial chemicals, or are they related to the food? But they also used a part of the plasma sample to test in in vitro assay to study the neurite outgrowth. So they looked for the individual chemicals, but they were also interested in the possible hazard on the neurite outgrowth. But in addition, they did some additional work with where they made these mixtures consisting of different components in order to and also studying this in the neurite outgrowth. But for me, the new thing is that they tested really the blood of the mothers in this. They made an extract, and the extract was tested in this neurite outgrowth, and then they concluded that the effect they see on the neurite outgrowth is stronger than you would have expect from the individual chemicals. So there is indeed a mixture effect on the same target.
Okay, much more clearer now, at least from my mind. Thank you. So I see another question here from Martin. So how far do you think we are in the determination of an actual human exposure description of micro and nanoplastics? Question mark. So there are many issues in regard to quantifying the exposure in a biological matrix, and we see a lot of data coming out that might be might not think.
I
Yeah,
think
no, yeah. You felt away at least at my end, Peter.
Okay, sorry, but
Yeah,
you can.
Raymond can also answer this probably, but yeah, we see all these publications about micro and nanoplastics that are detected in all kinds of tissues, in placenta and in brain and in penises and in I don't know what. Yeah, my response to this is it's at this moment we lack analytical methods to really for the nanoplastics, the plastics that are less than 1 micrometer. We don't have good analytical methods to detect these plastics. And these plastics are of 1 micrometer because these are the plastics that can cross biological membrane. So we should be very interested in this. And I also think that a lot of the concentrations and the total amounts of plastics that are reported, yeah, you have to be very careful when you do these analytical methods because you really need to do it very well and very controlled and avoid contamination of your sample during the collection and the processing and the analytical procedures. So we yeah, you see more and more that people are really questioning of the quality control of the micro and nanoplastic analysis. I think there should be much more rigid standards how to do that, because some of the values that are reported, when we go back to this Bradford-Hill criteria about biological plausibility, sometimes the values that are given cannot be true. You know, you cannot have in the publication that that has been circulated about levels in the brain. These levels cannot be true because then there are too much
There's
plastic
no
in your
brain
brain.
left anymore.
No way. Yeah.
Okay, Peter, did you have any comment on this? Raymond, sorry. We don't hear.
I agree with Hubert, of course, because in the same project we encountered all these problems with analysis in tissue. I mean, it's a big challenge.
Yeah.
And we work on it, but I always see from the chemical analytical people how difficult it is.
Okay, so yeah, thank you. I've got here a question in the chat, but I see it's the same question as in the Q&A. So Constantina saying that thank you for your interesting talk. What are the primary MIEs at your IOP derived from mixtures linked to hepatotoxicity in relation to nanoplastics?
Well, it depends, of course, which hepatotoxicity you're studying. You know, are you studying steatosis or other effects? So yeah, for the liver we have quite a number of AOPs. We have quite a lot of molecular initiating events presented, and it depends on the affinity of chemicals for the receptors in the molecular initiating event. So this cannot be easily answered, but I think it's very good that we have this AOP network, that we try to organize mechanistic data in these AOPs, and for the mixture assessment, it's really important to know this, to understand this molecular event, which one is which affected, because then you can say something about the combination effects.
Okay, yeah. I got just a small remark or a question on what you were saying at the end on, the different plastics to look at and what leaches from them is very important. But I learned from the talk of Raymond that, it's not only the leaching, but also what adds up on the plastic probably makes a difference, as for instance LPS from the Rhine River. So when they are exposed in the outside, And also contaminants from the environment, or maybe even metals, can also be quite important when we really look at microplastics. But yeah, that's not the pure product as it is being produced. I realize. So it's a difficult and an additional step to add to it. But what is your view on that?
You asked me, right? Sorry.
No.
Yeah. Raymond, you can answer.
Yeah, well, that's the complexity also. We are not exposed to primary particles mostly. We are exposed to particles in our diet or in our in the air we breathe, and none is sterile. Our gut is not sterile, the lungs are not sterile, so it's always combined exposure. And that also applies to all the other chemicals. So if you think about the immune system, which of course is susceptible to all these microbial components, but not only the immune system, but in particular the immune system. So if you look at the immune system, it's always complex because you have a combined exposure to microbes and chemicals. And you cannot separate it. And that's not also what we need. I mean, we need real life exposure data, real life risk data. And that's clear to all of us. I think that's also what Hubert says, that we are interested in and we all for combined exposures. That's the real life situation. And all the testing you do with single chemicals in a clear environment is nice for mechanistic reason, but not relevant for risk assessment. It is relevant, but not complete data.
Okay, yeah. Oh yeah, I agree with that. So we really need more holistic approaches in our exposures. Yeah. But we are missing that really. Yeah.
Yeah, and it's also difficult. I mean, that's why we always didn't do it in the past, because we didn't have the opportunities. Now with all the opportunities of combined exposure, data analysis, AI maybe.
Yeah.
Now we are at the point that we may be able to study that.
Okay. Yeah. It's maybe a good conclusion for this afternoon. That it's an exposome symposium anyway. So yeah, holistic approach in exposure and health effects is probably key if we want to progress. And certainly that paper from Brown really shows that small exposures seems to be innocent until you combine them, and then it is getting an issue. So yeah. So I don't see any things in the chat popping up or in the Q&A. So I thank my three speakers for sure. Raymond.
Thank you.
Also, sorry, Xavier. I was thinking of your second name, Basagaña. I don't know if I pronounce it right, and Hubert. So thank you very much for this very broad scope, but interesting view on what is exposome and how complex it is. I also have to thank Rani and Julia to help and assist in having this getting organized. And so thank you very much, and also my public for sure. I do not see you and how many people you are, but it's very nice to have at least the public. We had about 100 subscriptions for this, so that is substantial. And thank you for listening in. But I hope we did not bring horror at the evening of the horror of 1st of November. But anyway, I wish you a nice, for most of you, probably next day holiday. So see you, and maybe we will have another opportunity to have a symposium on exposome. Thank you. Bye-bye.
Bye.
Thank you. Bye-bye.